Good morning, everybody. I am really sorry not to be with you today. I hope that this record will fulfill your expectation about the subject Analytical Method Lifecycle Management. Please let me introduce myself. I am Gérald de Fontenay. I am an analytical chemist in charge of analytical services at AmatsiGroup. This group was newly created in 2013 as a venture between four different companies. It is based in Europe and North America and provides services to the pharmaceutical industry, services for product development, manufacturing, and supply of clinical batches. Regarding the analytical part, development, validation, transfer of analytical methods represent 20% of our activities, and using methods for routine QC testing or stability studies or some investigation represent 25% of our business. 16 years of experience serving the pharmaceutical industry provided us a strong knowledge about analytical methods and the management of their life cycle. So let's go to the presentation now. Analytical methods are used to generate data that will provide proofs about the quality of your product. What is quality is certainly a good question, but I guess you already know a lot about it. At least you should. The quality of your product, anyway, does depend on the manufacturing process, of course, but also it will depend on the image of the quality that is mainly provided by the analytical methods through the results generated with these methods. The results are going through an interpretation process that will lead to decisions regarding your product or your manufacturing process. So both the manufacturing process and the analytical methods should be mastered. Indeed, as it could be understood in ICH Q8 guideline, the knowledge about your process and your product is directly depending on the reliability of your analytical results. It means to the performances of your methods. Unfortunately, it is like the chicken and egg or egg and chicken paradox. You need a good method and reliable results to know more about your product. But you need to know your product well in order to develop methods with performances in line with the intended use of this method. Well, what does it mean in the real life? It means that the formulation development team and the analytical development team will have to work closely together all along the development steps of your product. When we have a look into these development steps, we can see that we will start at the early beginning with generic methods in order to acquire the first information about your product. Then we will shift to more specific and stability indicating methods during phase 2 and phase 3 to get all the required information on your product. At the end of phase 3, before market authorization, additional efforts are needed to select and optimize all the methods that will be required to prove that your critical quality attributes are well mastered before and during the commercialization phase. The definition of the right control strategy will then include the final methods that will need to be QC friendly. That means as simple and robust as possible. Here we have another point of view about these product development phases, what we expect as an evolution from one phase to another. Depending on the actors of the pharmaceutical industry, the goals may be very different, in fact not that much. You will have people who are following scientific drivers in order to increase product knowledge during the development steps, especially during the early phases. Other people will be more involved in reg affairs and they will need insurance that the methods are fully validated before the market authorization step. And we all know that the business people will require a cost as low as possible for the processes involved for your product, including these analytical processes. All of these scientific, regulatory, and business drivers have to be in mind of the analyst at the bench or around them. This is the right state of mind to reach your goal in the most efficient manner. If we go back to the subject, the method life cycle, the main steps are method selection, development, validation, and at the end, a routine testing. But nowadays, additional steps are described, if not required, in order to avoid shortcuts that could impair the efficient management of their life cycle. First of all, pre-validation steps, including robustness studies that are now required, as described in the latest FDA draft guidance about method validation. After the validation step, transfer methodology are now described in USP Chapter 1224, and this step is required in the new GMP Chapter 6. During routine use, some flexibility about method change control can now be expected if the method is well mastered, well understood, if a design space is clearly defined. Well, let's go deeper into the different steps of this life cycle. First, selection. We have to select methods that are adapted to their intended use. According to the egg and chicken paradox, the selection process will go back and forth in order to gain knowledge about your product, and then to adapt your methods to know even more about the same product. What is important at the end is to think about the final user with the scientific, regulatory, and business drivers in mind. Development step. Well, to obtain what I call a sustainable analytical development, you need analytical skills, of course, but you should never forget the word chemistry in analytical chemistry. Some may sell you development and optimization softwares as a magic potion. Such software can help a lot, we will see that later, but it will never be self-sufficient. The chemistry of the compound of interest will always drive your development, and it will also explain most of the impurities you will have to detect and analyze to characterize your active substance or, later on, your drug product. Some statistical skills are also important to understand measurement uncertainty, to motivate your analyst to decrease all the potential sources of variability during the development steps, and also being able to define clever design of experiments will also help you to set up useful robustness studies. Well, I just said that the optimization software are not sufficient, but for some tricky methods with several peaks to separate, they can be really helpful to design an efficient and robust method. In this example, if we focus on the separation of a critical pair of peaks here, we can define an orange zone here, where the resolution between this peak will always be in line with your expectation. On the contrary, if your analytical methods are close or near a cliff, near a dark zone, somewhere here, you may get into trouble during routine testing and fall from this cliff. This can generate a lot of difficult investigation and an overload of paperwork if you do not master completely the method before you use it for a routine test. Mastering before is sustainable. An important part of the method evaluation to be performed before the validation is the robustness study. It will describe the forces and weaknesses of your analytical method and will define the changes that do not impact the reliability of your analytical data. These robustness studies are really a cornerstone for potential regulatory flexibility. At the end of development, you will have defined a control strategy within a controlled space here. Wherever you are in this space, your method is reliable. This control space is part of a design space, which is also included in your knowledge space. But to be realistic, you will still have an unexplored space uh, because nobody can test everything. But what is important here is the size of your control space that brings regulatory flexibility later on. After development, a pre-validation is a useful step that can help you to define precisely acceptance criteria for your validation protocol, criteria that would take into account the real method performances and your product specification. This validation step becomes a simple confirmation required by reg affairs, as they need to prove that the method was validated. But the most interesting part of the validation exercise is to prove that the method is really valid from a scientific point of view. A good way to prove that it is really valid is to go through an analytical risk analysis using the validation results. You have here a graphical evaluation of the levels of risk you take according to the validation results you obtained. The risk levels that are presented here are corresponding to the risk of generating a false negative out of specification data for the assay of what I call a perfect batch with an actual content of 100%. So this out of spec result is only due to the method itself. You have a 5% risk as a red dotted line here. This risk level is the limit that could be accepted. You can easily plot the risk level for your own analytical method if you take into account the accuracy results on the x-axis here and precision results on the y-axis here. For accuracy, a 0% bias corresponds to a 100% accuracy result. 2% bias would correspond to 98 or 102% as an accuracy result. For precision, you simply plot the RSD percentage on the axis. As an example, we have method 1 here with a very low bias, so accuracy is close to 100%, but you have a 3% RSD, which is quite high. If your acceptance criteria goes that high, you may consider that this method is valid, but during a routine test, approximately one result out of 10 would be out of specification. That means below 95 or above 105% for a batch with a natural content of 100%. So I don't know if you can accept such a risk. I wouldn't. As another example, under the arrow there, it is more or less the same. For this method, you have an RSD of 2% and a bias of 2%. So you are at the limits, but still within classical acceptance criteria with an RSD for precision at 2% or below, just below, and your accuracy result between 98 and 102%, even if it is at the limit. Here, again, the risk of generating out-of-spec results due to method performances only is still above 5%. The best, of course, is to see your method in the green zone, below the 5% limit. It is certainly better to see it below the 1% limit, I guess. You will find a reference regarding the article with this kind of curves at the end of this presentation. I can, of course, send an electronic copy to you by email if you ask for it. Let's go to method transfer. As explained earlier, the transfer became a mandatory step. Again, from a scientific point of view, the aim will not to have a nice transfer report, but more to prove that the results and their reliability are not depending on the laboratory where they are generated. The way you manage such transfer can vary from one lab to another, from one situation to another, But as soon as you prove that the data at the receiving lab are reliable enough, you have performed a nice method transfer. Of course, you have to document it. At the end, we reach the routine testing, a good QC release testing. In this case, control charts are really an efficient quality tool to prove that your method remains valid. You can see on this slide some charts related to method system suitability test results, really related to your analytical method. When you consider your charts with batch-to-batch assay variation, it is slightly more difficult to manage as they represent the sum of both method and process variability altogether, and it's not easy to separate what is due to the method and what is due to the process. I would suggest anyway you to include in your annual product review a review of these control charts in order to ensure that the quality of your product is not impaired by unexpected changes in your manufacturing process and or in your analytical procedure. Unexpected changes, as soon as we talk about changes, the unexpected ones are indeed the most dangerous sources of trouble regarding your product quality. This is why the monitoring of your method is so important. May I also suggest you to anticipate as much as possible this kind of unexpected changes? The best example I could provide to you is about the potential evolution of your product during its shelf life. For example, a cream could become thicker and then the recovery of your active substance could decrease during a stability study due to the evolution of your formulation. This could be avoided or anticipated at least if you include during earlier development steps the testing of stress sample of drug product instead of going directly to the performance of a classical validation study which is generally performed with fresh samples only. This simple example illustrates the importance of thinking about the final users during the early development steps. For the top of the page, the deliberate changes, it is much easier to manage. You can compare before, after the change and analyze the impact of your change on an analytical method. As soon as it is well documented, your method is well managed. I could not end this presentation without a word about the new trends for method lifecycle management. Performing an analytical method is really a process like others. And the current changes about process validation can also be applied to analytical methods. An initial IQ, OQ, PQ qualification could replace a classical validation exercise in order to prove that ATP, which means analytical target profiles, are met. Meeting the ATP means that the method is adapted to its intended use. You define the use and you see that your method is adapted for that through the risk analysis. Qualification could then be performed as often as needed during the product life cycle as a continuous method verification during a change control, during a transfer, and so on. Such a way of thinking is fully in phase with my own thoughts discussed today. It is not because a method has been validated before that it is really valid, nor that it is still valid. The reliability of your data should be proven all time long during the life cycle of your own product. I hope you enjoyed this overview and you will see differently your analytical methods now. And to go deeper into this subject, I selected some official or literature references for you. I really thank you for your attention and please do not hesitate to contact me if you wish to continue a discussion about the subject. Again, I am sorry not to be there and to be unable to respond to your questions today. Sorry not to be in Spain because I love Spain, and I hope to meet you during another another Congress, perhaps CPHI in Paris or AAPS in San Diego this year, or another GMP Summit next year. Bye bye.